Alright guys, so now we're going to cover section 12.3. We're going to do a survey of the major antimicrobial drug groups. I think we've talked about this concept before, but there are sort of two ways you can define an antimicrobial, and that's whether it's a broad spectrum versus a narrow spectrum. So broad spectrum drugs are really going to be effective against more than one group of bacteria, or in some cases, eukaryotes as well. One example is tetracyclines. So tetracyclines are good to give, obviously, if you don't really know what the causative agent is, and that is, the idea would be you could kill off a wide spectrum of bacteria. The narrow spectrum drugs are actually more ideal. These guys are only going to target a specific group of microbes, and so they're more pinpointed against the microbe. Two examples we'll talk about are polymyxin and the penicillins. Okay, so this is a good table that talks about the spectrum of activity for the different antibiotics. So here you can see tetracycline that we just talked about and how broad it is. I would say probably the carbapenems is going to be your second broad class. Those are really good against both gram-negatives and gram-positives, and even a little bit it can be used for mycobacterium. And actually tetracycline, as you can see, is the drug choice if you do have Rocky Mountain spotted fever. Polymyxin is one of those that's very narrow in scope and really is only good against some mycobacteria and a few gram-negative. All right, so the first class of drugs we'll talk about are the penicillins. So these are the classic class of drugs that target the cell wall. And essentially, this is a group. These originally were naturally derived, but now they can be synthesized in the lab. And here is sort of the basic structures of some of these penicillins. Here are the different names: napcillin, cloxacillin. So essentially there's three main parts to any of the penicillin class of drugs. So this yellow structure here is the thiazolidine ring. In red is the beta-lactam ring. And then over on the side is the R group, the variable change, which is the one that really changes for the different penicillin classes. All right, so again, the way penicillins work is they target the cell wall. Because of that, the cell is the bacterial cell that it targets lyses because that cell wall is no longer intact. And so here are some of the subclasses, subgroups of the penicillins. Penicillin G and penicillin V are the most natural sort of forms. They're mainly prescribed because they are very effective against the gram-positive cocci, but they have some effect on gram-negatives as well. If you recall, gram-negatives also have a cell wall, so they can be effective against those. Ampicillin is a class that we use in the lab. This particular class is semi-synthetic, and it's usually used against gram-negative enteric rods. So if you recall, this is one we use to kill off E. coli. And then there's some more penicillin subgroups. So this particular chart is then just going through and naming the different types of drugs, defining whether the spectrum of action is narrow or broad, and what the particular use or advantage would be, as well as disadvantages, such as poor absorption, it causes allergies, easily hydrolyzed by the penicillinase, other disadvantages. So the cephalosporins are sort of the second class of drugs that were introduced that target the cell wall. These were a natural isolate from the organism Cephalosporium acremonium. They as well have a beta-lactam ring, which is shown here in red again. And essentially this ring can get chemically altered. So chemists have gone in and changed this. Essentially, the mode of action for the cephalosporins is the same. It will target the cell wall and cause lysis. All right, so in this particular table, they're going through the cephalosporin drugs. Essentially, what it does is it lists the first generation, second, third, fourth, and the fifth generation. So essentially, these were the original cephalosporins that were used. Cephalothin and cezazolin. Nowadays, now that we have methicillin-resistant Staph aureus, we have a fifth generation of drugs, and so now the common cephalosporin that's prescribed especially against MRSA is ceftobiprole. Okay, so if you notice, it's easy to recognize the cephalosporins. They all do start in CEF. Okay, another class of drugs that target the cell wall is the carbapenems. So these are very good at breaking up the cell wall. These two subgroups, unfortunately, are very powerful, but they're really toxic. So essentially, these drugs are used as sort of a last case scenario when other drugs are not working. And then these are some miscellaneous drugs that target the cell wall. So bacitracin is one that you may have been familiar with. It's the main ingredient in Neosporin. Neosporin actually has a few antibiotics in it, but bacitracin is the main ingredient. And it's very good at combating superficial skin infections. Isoniazid is a common drug that targets the cell wall, and it's powerful in combating tuberculosis. And then the last one you may have heard of as well, this is vancomycin. Vancomycin is also a drug that's sometimes chosen in cases where there's allergies to penicillin or there's methicillin resistance in certain patients. All right, so we'll move on to some drugs now that target protein synthesis, okay? So the first class we'll talk about are the aminoglycosides, okay? So the aminoglycosides are composed of one or more amino sugars and then an aminocyclitol ring. So this is the aminocyclitol ring and 2 amino sugars. So these aminoglycoside drugs are the products of a class of microorganisms called the actinomyces. Here is what a typical actinomycete looks like. And the aminoglycosides have a very broad antimicrobial spectrum. All right, so this is an up close and snapshot kind of different types of drugs and how they affect the bacterial ribosome. So, I won't go into too much depth. Obviously, you can look into this. I'll point out a few of these, but these are, 5 different ways in which the ribosomes can specifically be affected in different ways by 5 different types of drugs, but they all target protein synthesis. In particular, I'll just point out the aminoglycosides. Okay, so here is your 50S subunit, the 30S ribosomal subunit. Translation is occurring, but for some reason, when this drug interferes, the messenger RNA gets misread, and so the outcome of the protein then, as we all learned, is derived from the messenger RNA, and so then all of these proteins then obviously get translated wrong. Okay, so the subgroup that you're probably most familiar with, you've heard of before, that's an aminoglycoside is a streptomycin. Again, it's a very broad spectrum used to treat infections by both gram-negative rods and gram-positive bacteria. So this is probably the most common aminoglycoside that you've heard of. Okay. Another type are the tetracyclines and the newer derivatives called the glycyclines. So the tetracyclines are a natural parent compound, but there's also synthetic derivatives. They have, again, if you remember that chart that we showed before, it's got the most broad spectrum of all of the antibacterial drugs out there. Unfortunately, it does have some side effects, GI disruption, probably some diarrhea, and then deposition into hard tissues. This newer derivatives of tetracycline also target protein synthesis. Specifically, they block the 30S ribosomal subunit from interacting with the 50S ribosomal subunit. Effective against bacteria that have become resistant to the tetracyclines. All right, so this just talks a little bit further about the tetracyclines, the specific action. This looks like it is a misprint here. This should be drugs that target the protein synthesis, not the cell wall. So tetracycline is the most common, and tigecycline is another type of derivative of tetracycline. Erythromycin, another targeting antimicrobial for protein synthesis. This particular structure has what's called a macrolide. You guys do not have to know what the structure of that looks like. Very broad spectrum, and the good thing is it does have some low toxicity. Clindamycin, another one you may have heard of before. Unfortunately, there can be some adverse reactions with the GI tract. So erythromycin, as well as clarithromycin, azithromycin, these are all subgroups of the macrolides. Again, these are drugs that target protein synthesis. See, the azithromycin is commonly used in AIDS patients as well to fight off the mycobacterium that's a common infection in AIDS patients. And here's a few of the miscellaneous drugs that target protein synthesis. I'll just mention linezolid just because it is sometimes used to treat antibiotic-resistant organisms such as MRSA and VRE. So the last class we'll talk about are folic acid synthesis targeting drugs. These are the sulfonamides or sulfa drugs. And these are the first modern antimicrobial drugs. They are synthetic. So these were completely synthesized by scientists. No natural products were looked at or had an origin in them. And then here's just a few of the types of sulfonamides you might run into. So essentially what the sulfonamides do is that they interfere with folate metabolism by blocking specific enzymes required for this tetrahydrofolate synthesis. The tetrahydrofolate is needed for cells because of folic acid synthesis, which is integral for DNA, RNA, and amino acids. So this is kind of the mechanism of action of how targeting folic acid synthesis can kill off microbes. Okay, so we're going to take a break there, and I'll come back in the next section to finish off section 12.3.